Hello dear learners. In this particular presentation, I am going to give a talk on the topic of mutations. What is the meaning of mutation? It is a sudden heritable changes that is caused in the genetic material. It is known as mutation. The term mutation refers to both to change in the genetic material to the process by which the change in the genetic material is occurring. If there is no mutation, what will happen without mutation? All genes would exist in one form, alleles would not exist, genetic analysis would not be possible, it won't be able to evolve and adapt to environmental changes. Mutation can alter the DNA sequence. At molecular level, mutations are alteration in the DNA molecules. Chromosomes can exchange parts and genetic materials that can even jump from one to another chromosome. Mutation on the basis of inheritance can be divided into somatic mutation and germline mutation. Somatic mutation is occurring by the division of cell. If a cell is having some mutant property, when it divides, it carries the property to the next thing. Such as a normal cell, a chemical mutant is acting on it. Okay, so initiation of the mutation takes place, then see, this is the mutated cell, then promotion takes place, this is the mutated cell, then the cellular division takes place and adoption of cell toxicity happens. Okay, cell proliferation leads to cancer. So this is how somatic mutation occurs. In germline mutation, the change occurs during the DNA replication, the process of meiosis. The resulting gametes and all the cells that descend from it after fertilization have the mutation. Okay, so it is mainly affecting the gametes during the process of meiosis, and once the mutation has occurred in the step of cell division, the mutation is carried out in the fertilized gametes also. Afterwards also, after the fertilization also, the offspring are carrying the mutated property. That is the definition of germline mutation. What is the classification of mutation? There are different ways of classifying the mutation: functionally, structurally. Functional classification is harmful, beneficial and neutral. Structurally, there is the point mutation, frame shift mutation, silent, missense and nonsense mutation. We start with the harmful mutation. Mutation can cause errors in protein sequence, creating non-functionally proteins. Harmful mutation can cause genetic disorders. Drosophila melanogaster, a Drosophila with legs grown over the head as a result of a mutation. A baby with an abnormal formation of eyelids because of the mutation. So, this type of mutations are harmful mutations. Okay, here you can see the sixth finger is there. So, these are the harmful mutations. We are going to focus on beneficial mutation. It is the ultimate source of all genetic variation, raw material for evolution and able to evolve and adapt to environmental changes. Here I have cited one example of beneficial mutation: protection against HIV infection. See, what is the general phenomena of HIV infection is that there are cytokine receptor 5 in the body. To enter the cells, one virus of HIV has to attach itself to the CCR5 receptor on the cell surface. Only then it can penetrate inside the cell surface. If someone is born with a mutation in which the CCR5 receptor is not there, that person is automatically having a protection against HIV. How? Because it cannot allow the HIV strain to attach to the receptor, and if it is not attaching, it will not insert inside the body. So it is called as a beneficial mutation. Neutral mutation. It is that type of mutation that occurs in an amino acid codon but has no impact. Here I have cited one example. There is a change in the sequence of amino acid in this particular site, okay, since strand of mRNA. But this change in the base pair results in an amino acid change, but the new amino acid has the same chemical properties as the old amino acid. So this is called as neutral mutation. Here you can see the different types of mutation falling under the category of structural classification. We'll start with the point mutation. A point mutation or a single base substitution is a type of mutation that causes the replacement of a single base nucleotide with another nucleotides of DNA or RNA. In the diagram, you can see wild type hemoglobin DNA CTT, only the single T is replaced by A. The T nucleotide is replaced by A. Similar manner here in the mRNA, the GAA, the mRNA, the central A base is replaced by the base U. Okay, so this is the point mutation. DNA structure permits some basic type of alteration or mutation at a site. That point mutation can be categorized as transition, deletion, insertion, transversions, translocation and inversion. Okay. Deletion is, you can see here that A, B, C, D, E, F, G, this is a hypothetical base pair, A, B, E, F, G plus C, D. C, D is lost, it is deleted from here. Insertion is A, B, C, D, E, F, G, C, D, we add up the, insert the extra C, D pair, A, B, C, D, C, D, E, F, G. So, one C, D is inserted. Inversion. Inversion is A, B, C, D, E, F, G, A, B. What is the change we have made? We have inverted the position of C and E. Okay. E has come to the position of C and C has gone to the position of E. Translocation is, translocation are a transfer of a piece of one chromosome to a non-homologous chromosome. Translocation can alter the phenotype in several ways. The break may occur within a gene destroying its function. Translocated genes may come under the influence of different promotes and enhance so that their expression is altered. The breakpoint may occur within a gene creating a hybrid gene. This may be transcribed and translated. Okay. So, this is a normal chromosome 8, this is a normal chromosome 14, okay, and this has been translated to chromosome 8q minus and chromosome 14q. See here, the changes have occurred here. Okay, at this point, the color changes, I hope you can see it. Okay. What is transition? The replacement of a purine base with another purine or pyrimidines with another pyrimidine is called as transition. So, a purine base, when it is replaced with another purine base or a pyrimidine base which is replaced with another pyrimidine base, that is called as transition, and transversion is replacement of a purine with pyrimidines or vice versa. Okay. Here purine gets converted to pyrimidine or pyrimidine gets converted to purine. So, here diagrammatic representation is there. If you go through the diagram closely, you will be able to find out the difference between the transition and transversion. Next we will discuss about missense mutation. A point mutation can also change a codon so that a different protein is specified. A non-synonymous change occurs. For example, sickle cell disease. Okay. So, DNA transcription and RNA translation with the help of a protein hemoglobin A, normal red blood cells are produced. DNA transcription and RNA translation protein hemoglobins, sickle cell hemoglobin means sickle red blood cell are produced. What is the change you can see? You can see the change here. This is the point of mutation. If this point mutation occurs, then you will see that the normal red blood cells, normal RBCs are not produced. Instead of that, a sickle red blood cell is produced. Nonsense mutation. A nonsense mutation is a genetic mutation in a DNA sequence that results in a shorter unfinished protein product. nonsense protein product, you can say. Okay, so during protein formation, the DNA nucleotide sequences are read 3 nucleotides codon, and each codon corresponds to a specific amino acid or stop codon, that is also called as nonsense codon. This nonsense mutation occurs when a premature or stop codon is introduced in the GNA. When this nonsense codon is introduced in the GNA, that situation is called as nonsense mutation. When the mutated sequence is translated into a protein, the resulting protein is incomplete and shorter than the normal. Most nonsense mutations result in non-functional protein. For example, blood clotting disorders, Becker muscular dystrophy, and Ehlers-Danlos syndrome. Sense mutation. Sense mutation are the opposite of the nonsense mutation. Here a stop codon is converted into an amino acid codon. Okay, just the opposite of the nonsense. There an amino acid codon was converted to a stop codon. Here the opposite thing is happening. Since DNA outside of the protein coding regions contains an average of 3 stop codon per 64, the translation process usually stops after producing a slightly longer protein. For example, hemoglobin alpha constant spring alpha globin is normally 141 amino acids long. In this mutation, the stop codon UAA is converted to CAA, that is glutamine. The resulting protein gains 31 additional amino acid before it reaches the next stop codon. This results in thalassemia, a severe form of anemia. I think you all are aware of the term thalassemia. Frameshift mutation. Frameshift mutation is a genetic mutation that is caused by indels, that is insertion or deletion, that is indel, okay, of a number of nucleotides. Due to triplet nature of gene expression by the codon, the insertions or deletion can disrupt the reading frame. A frameshift mutation causes the readings of all codon after the mutation to the different amino acids. The protein being created could be abnormally short, long, or contain the wrong amino acids and is functionless. For example, Tay-Sachs disease. This is an example of frameshift mutation. What are the different causes of mutation? A mutation can occur spontaneously to be induced by chemicals or radiations. Spontaneous mutation on the molecular level includes tautomerism. In this, a base is changed by the repositioning of an hydrogen atom. Okay, here the reaction is shown to you, and a hydrogen atom is repositioned here, that is why the base is changed. Okay, you can see here this forms, this OH is shifted, NH1H from NH2 has gone there. Similar way here the NH is gone to the OH. So there is a change of the hydrogen atom position. NH2N was there, now it has shifted to NH. Okay. Spontaneous mutation, that is depurination. In this, the loss of purine base A or G occurs. Okay, deamination. Here is the example of your depurination. A purine base is lost here. You can see very clearly this base is lost. Okay. Deamination is changes a normal base to an atypical base, that is C changes to U. This is a cytosine structure. Via deamination you can see it has become uracil. How? By the change of this particular group. Deamination means amino group is removed. Induced mutation. Induced mutation are those resulting from the exposure of organism to mutagenic agents such as ionizing radiation, UV light, or various chemicals. Induced mutation on the molecular level can be caused by the chemicals such as hydroxylamine, base analog, alkylating agents, DNA intercalating agents like ethidium bromide, DNA cross-linkers, oxidative damage, nitrous acid, acridine dyes. These are the structures of the different types of chemicals that can induce the mutation. The name of the chemicals I have already mentioned in the earlier slide, and here I have mentioned the different structures of the alkylating agent, nitrous acid, acridine dyes. Okay, you can just go through these structures. Radiation, for example, X-rays and ultraviolet rays. Ionizing radiation breaks covalent bonds including those in DNA and is leading cause of chromosome mutation. Ionizing radiation has a cumulative effect and kills cells at high doses. UV radiation ranges from 254 to 260 nanometer can causes the purines and pyrimidines to form abnormal dimer bonds and bulges in the DNA strands. Okay. Mutational hot spots. In some genes, the mutation are more likely to occur in the region called as hot spots, where sequences are repetitive. You know the meaning of hot spots, the where the ability is more. Okay. So, where the sequences are repetitive, it is as if the molecules that guide and carry out the replication become confused by short repeated sequences. This is the region prone to mutation. For example, more than one-third of many mutations that causes alkaptonuria occur at or near or more CCC repeats, even though this repeats accounts for only 9% of the gene. Okay. Such a segment of DNA with identical sequences but opposite in direction is called as a palindrome. A palindrome site is a sequence of base pairs in double-stranded DNA that reads the same backwards and forward across the double strand. This is an example of 3' and 5'. Palindrome I have already explained in the earlier lecture videos also. With this, I complete my presentation and talk on the topic of mutation. I hope the concepts are clear to you. If you have any queries, you can ask me in the comment section, or you can also drop the queries in the Google Classroom platform. Thank you.